The overall goal of this procedure is to extract lipoproteins from bacterial cells for direct applications, and to further prepare N-terminal lipopeptides for structural analysis by MALDI-TOF mass spectrometry.
This method can help answer key questions in the immunology field, as a lipoprotein's N-terminal structure influences toll-like receptor recognition and signaling, impacting the host's immune response. The unique advantage of this technique is the optional but intentional formation of sodium adducts, which promotes fragmentation towards the diagnostic dehydroalanyl ion, aiding in structural assignment of the lipoprotein's acylation state.
Grow bacteria in 15 milliliters of tryptic soy broth or similar rich media to late exponential phase. Harvest the cells by centrifugation before washing once with tris-buffered saline EDTA or TBSE. Resuspend the cells in 800 microliters of TBSE with 1 millimolar PMSF and 0.5 milligram per milliliter lysosome. Transfer the solution to a 2.0 milliliter threaded microcentrifuge tube with screw cap and O-ring prior to incubating for 20 minutes at 37 degrees Celsius. Add approximately 800 microliters of 0.1 millimeter zirconia silica beads to the tube. Then disrupt the cells by shaking at maximum speed on a homogenizer for 5 cycles of 30 seconds each, with a 2-minute rest on ice between each cycle. Centrifuge the sample at 3000 times G for 5 minutes at 4 degrees Celsius to pellet beads and unbroken cells.